The ErbaMolecular RT-PCR assay is highly sensitive. It's a duplex assay with a limit of detection of 200 copies per mil. This has been demonstrated in nasopharyngeal swabs, oropharyngeal swabs, and saliva samples. We also perform regular genomic surveillance, and this is to check for any mutations in variants of concern to ensure that our assay is still able to detect them. With all this in mind, this allows us to deliver accurate results for our patients. The ErbaMolecular RT-PCR assay is highly specific for SARS-CoV-2. It's a duplex assay consisting of the N1 and RDRP targets, and these have been demonstrated to only detect SARS-CoV-2. We've tested these against other respiratory flora and microorganisms, and also other coronavirus, and these have been shown to not be detected by our assay.
The ErbaMolecular RT-PCR SARS-CoV-2 assay is a duplex test that targets the N1 and RDRP regions of the SARS-CoV-2 genome. The benefits of having a duplex test is that it offers increased specificity, and it's also beneficial in the case of gene knockouts that may occur in the case of variants.
The ErbaMolecular RT-PCR assay is very easy to use. It consists of 3 components: a lyophilized RT-PCR master mix, a positive control, and a dilution buffer. All you have to do is add the dilution buffer to the lyophilized master mix and the positive control, and then it's ready to dispense. The ErbaMolecular RT-PCR assay is highly flexible. It's been demonstrated to be compatible with different commonly available extraction workflows and also RT-PCR instruments. We've also demonstrated that the RT-PCR assay can be used with both automated and manual workflows to make this easier and more open to different methods within different laboratories.
Distribution of the test is handled completely out of the cold chain, meaning that it can be transported at room temperature, avoiding the need for dry ice shipment, making it safer, more efficient, and easy for global transportation, with the confidence that the test remains robust on arrival to our customers. With the ever-changing nature of the pandemic, there can be real issues in terms of lab managers planning for stock control. The longevity of the shelf life means our customers will have the confidence to obtain accurate, reliable results during the changing phases of this pandemic.
Sample pooling is a really useful method to improve the throughput of laboratories when processing patient samples. This is a benefit to the lab because it allows you to reduce your reagent costs as well as your extraction costs. And what this means is you can then process 5 patient samples with just 1 extraction and 1 PCR reagent mix. The advantage of this is as we move towards the end of the pandemic phase, when prevalence is hopefully going to start dropping, then pooling really will play a part in this and allow more samples to be tested and to still try to control the spread of the pandemic as best as possible. The ErbaMolecular RT-PCR assay for SARS-CoV-2 has a OR logic for target calling. What this means is that detection of either the N1 or the RDRP target is sufficient to call a positive result. We're confident that the detection of a single target is sufficient for a positive result, and we've demonstrated this with substantial sample testing. Now, why is this important? This is advantageous compared to other assays which require all the targets to come up positive because that's more vulnerable to mutation knockout. So if a particular target is mutated in a variant of concern in the future, this might mean that an actual positive result isn't called as positive. Whereas we believe by using detection of either target, this won't be the case. We recently published a paper in Nature Scientific Reports on the ErbaMolecular RT-PCR assay for the detection of SARS-CoV-2. This paper demonstrates the validation of the assay and also demonstrates efficacy of pooling samples 1 and 5. We believe this is really important because pooling samples is an important method to reduce the reagent cost and also the labour cost when processing samples for RT-PCR. And the reason why we want to do this is to make it easier for our consumers to be able to accurately detect SARS-CoV-2, maintaining the sensitivity, whilst also making it more cost effective for them, certainly as we move towards the end phase of the pandemic.
Pooling requires highly sensitive assays. The ErbaMDx RT-PCR SARS-CoV-2 assay is a highly sensitive assay with an LOD of 200 copies per ml. Our paper, published in Nature Scientific Reports, demonstrates that the Erba test kit can be used for pooling with a 1 in 5 pooling strategy with NP samples. As per FDA instructions, we validated the Erba kit against an FDA emergency use approved kit, and these studies demonstrated 100% positive percent agreement and 100% negative percent agreement. These results compare with FDA instructions, which require only 85% positive percent agreement and negative percent agreement. Our paper concludes that the ErbaMDx RT-PCR SARS-CoV-2 assay can be used for a 1 in 5 pooling strategy, and the high sensitivity it offers gives us the confidence that we can detect SARS-CoV-2 even in low positive patient samples.
The ErbaMDx RT-PCR assay is a really great kit, and the reason for this is it's highly sensitive. We can achieve 200 copies per ml detection in nasopharyngeal swabs, oropharyngeal swabs, and saliva samples. It's also really easy to use, just a simple one-step addition of your dilution buffer to your lyophilized RT-PCR master mix. And as well as that, we've got a really unique feature that actually the test has been validated for sample pooling. And as we approach the end of the pandemic phase, this is going to be really important to make sure that we're still able to achieve detection of SARS-CoV-2 in those samples that are still coming up as positive, whilst also managing to reduce the costs of both the reagent and the extraction workflows for our end users.
The ErbaMDx RT-PCR SARS-CoV-2 assay is sensitive. It has an LOD of 200 copies per ml. This sensitivity has also enabled it to be used for pooling. And pooling enables us to keep our costs down, enables us to also spare resources in times when we need these resources most. The ErbaMDx assay is also a duplex assay, and it uses an O1 call logic, which makes it particularly useful in the case of gene knockouts that may be caused by variants. And finally, the ErbaMDx assay is flexible. This means that it can be used on open PCR and workflow extraction platforms.